Namaskaram. My name is S. Ashok Kumar. Welcome to my channel. Now, in this video, in this part, I am going to explain different factors affecting enzyme activity. You can call different factors affecting rate of reaction. Mainly, there are three factors affecting. One is, first factor is substrate concentration. The second factor is effect of temperature on the enzyme activity. Third factor, effect of pH on the enzyme activity. For example, if you take our body, temperature and pH are relatively constant. That is why the most important factor influences the enzyme activity is a substrate concentration. Now I am going to start explanation. Effect of substrate concentration on the enzyme activity. In simple way I will explain. For example, if you take a reaction mixture, that reaction mixture containing 50 enzymes. As we know, each enzyme containing one active site. One active site associated with one substrate. So, 50 enzymes maximum associated with the 50 substrate. To that reaction mixture, for example, if you add the 10 substrates immediately combines with the 10 enzymes. That time 40 enzymes are free. Then at a time 10 substrates convert into 10 products. Okay na? For example, increases to the 30 substrates convert into 30 products. For example, if you take the 49 substrates, convert into 49 products at a time. Here, this is I am calling as a phase 1. Here, what is the conclusion of phase 1? While I am increasing the substrate concentration, rate of reaction also gradually increasing. That is where initially rate of reaction is directly proportional to the substrate concentration. This is a first order reaction kinetics or enzyme kinetics, what we know, first order. After that, if I increases to the 50 substrate concentration, here 50 substrate concentration is nothing but the concentration of the substrate is equals to the concentration of active site of enzyme. That particular thing is called as a saturation point. Saturation point where there is no free substrate, there is no free enzyme. Otherwise, you can tell saturation point where enzyme giving the maximal velocity. Beyond that, enzyme cannot give. Why? Because the active sites are completely associated with the substrates. After saturation point, for example, if I increases the 60 substrates, out of 60, at a time 50 only convert into products. 10 substrates remains as substrates. For example, 90 substrates. Out of 90, 50 only converts into products. Remaining substrates like that. This is a phase 2. Here even I am increasing the substrate concentration, but rate of reaction remains a constant. That is why rate of reaction become independent of the substrate concentration. Now you can call this is a zero order reaction. That is why what you need to understand initially, rate of reaction is following the first order. After saturation point, rate of reaction becomes a zero order. The same what I theoretically explained comes to take the diagram. Here what happened? Rate of reaction or enzymatic activity versus substrate concentration. Here, initially when the substrate concentration increasing, rate of reaction gradually increasing. Yes or no? This is a saturation point where enzyme giving the maximal velocity. After saturation point, even though substrate concentration increasing, the rate of reaction remains a constant. This is a graph. Here the very important constant is this one, Km. Michaelis constant, Km. Km is a Michaelis. Michaelis Menten constant, that is important constant. Now first I will define what is a Km. Km is a substrate concentration where enzyme giving the half of the V maximal. Okay, now you have to remember Km is nothing but substrate concentration. So actually, what is the importance of Km? Why we need to remember the Km? That is, I am giving a or important relation. That is, if you know the Km value of an enzyme is inversely proportional to enzyme affinity to substrate. This is a very important relation you need to remember. Km is nothing but substrate concentration. Okay, na? Km value is inversely proportional to the enzyme affinity towards the substrate. I will make you understand now. For example, take one equation. Glucose convert into glucose 6-phosphate. Okay, na? This reaction is mediating by enzyme glucokinase or hexokinase. Okay, na? Now, for example, this glucokinase can combine with the glucose. Hexokinase also combines with the glucose. For example, here, if you take the glucokinase Km is equals to 10 millimolar of glucose. What is actually glucose? Glucose is nothing but substrate concentration. Km is nothing but substrate concentration. So, glucokinase Km is a 10 millimolar of glucose. For example, you take the hexokinase. Hexokinase Km is equal to 0.1 millimolar of glucose. With this one, you can tell glucokinase can combine with glucose, hexokinase. Now, I am asking you, among hexokinase and glucokinase, which enzyme is having the more affinity towards the glucose? That means, which enzyme is having the less Km value? That is hexokinase. No? That means, among hexokinase and glucokinase, what is happening? Hexokinase is having the more affinity towards the glucose than the glucose. But you need to remember, both hexokinase and glucokinase both having the affinity towards the glucose. I am telling hexokinase is having the greater affinity towards the glucose than the glucokinase. That is why here this relation you should remember. Km is nothing but substrate concentration where enzyme giving the half of the V maximal, that is inversely related to the enzyme affinity towards the substrate. Effect of substrate concentration on rate of reaction. Now, I am going to tell about the effect of temperature or enzyme activity. Is very simple. For example, take this graph. Rate of reaction or enzymatic activity versus temperature. Here, you can start 0 degree centigrade, 10, 20, 30, 40 like that, 10 difference. Here, enzymatic activity 0, 1, 2, 3, 4, 5 up to 10. Okay. Here, what is happening? Initially, when the temperature is increasing, rate of reaction also gradually increases, as we know. A particular temperature where enzyme showing the maximal activity is the optimal temperature. Optimal temperature for our enzymes is around 40 degrees centigrade. Around. Okay, now you understood now why we are maintaining 37 degrees centigrade. Because 37 degrees centigrade, we are achieving the maximum activity of the enzyme and getting the product. Even if the temperature, after optimal temperature, if the temperature is increasing 50, 60, enzymes are basically proteins. They undergo denaturation and loses the biological function. That is why you are getting the bell-shaped graph. Here, what is important, no? Effect of temperature or enzyme activity. There is a constant called as a Q10, temperature coefficient. What is a Q10 means? Temperature coefficient. For example, K35 by K25. For example, what it means is that if the temperature, K for Kelvin, if the temperature is increased from 25 degree Kelvin to the 35 degrees, means there is a difference of 10 degrees. When the temperature is increased, 10 degrees centigrade increase, the enzyme activity is increased around 2 times. Okay, for example, if the temperature 10 degrees centigrade or 10 degrees Kelvin, if I decrease, the enzymatic activity decreases 2 times. That is why this is important. What is a Q10 or temperature coefficient? When there is a change in 10 degrees centigrade or 10 degrees Kelvin, enzyme activity change around 2 times. So why I said around? Some enzyme activity change 1.5 times, some enzyme activity 2.53 times. That is why I said temperature coefficient or Q10 value is around 2. This point is important for the NEET exam. Next comes to the effect of pH is a normal thing only. When the pH is gradually increasing, enzymatic activity increasing, when it reaches the optimal pH where enzyme showing the maximal activity. Optimal pH is around 7. Means indirectly meaning what? It is around neutral only. Na? If the pH is increasing, automatically enzymes undergo denaturation, they loses the biological function. That is why optimal pH of most of the enzymes around 7. If you take the salivary amylase, what is the pH of the optimal pH? That is 6.8 as we know. Intestinal enzymes you can take. That succus centricus enzyme digestion. That pH is around 7, 7.4 only. I said not 7, around 7. There are some exceptions also there for this one. For example, exceptions if you take, for example, if I tell pepsin, what is optimal pH? 1.8. Yes, strong SD needing only pepsin get activated as we know. For example, you take lysosomal enzymes. Lysosomal containing the hydrolytic enzymes now. What is the optimal pH? That is a 4.5 to 5 SD pH now. That is why there are definitely so many examples are there around 7. There are definitely exceptions also there. These are the different factors affecting the enzymatic activity or rate of reaction. Till now, if you understand the concept, please like and share to your friends. If you have any doubts in the biomolecules topic, you can comment below. Definitely, I will respond to your questions. Thank you.